is an exogenous protease. By increasing the breakdown and solubilization of protein, ProAct supplements endogenous trypsin in the digestion of protein in livestock gastrointestinal tracts. The result is improved protein utilization and animal performance, reduced feed costs, and lower nitrogen emissions. Soy contains a range of anti-nutritional factors, including trypsin inhibitors. As their name suggests, they inhibit the action of endogenous trypsin enzymes. That results in negative effects on animal performance, including reduced protein digestion. Unlike endogenous trypsin, ProAct is not impacted by anti-nutritional factors in soy. Let's head to the lab for a demonstration of the effects of ProAct on soy and a comparison of the effects of trypsin inhibitors on endogenous trypsin and on ProAct. We start out with milled soy. We treat the soy with a blue dye that attaches to proteins, coloring them blue. We separate the soy into 5 vials, then add buffer solution to all 5. The buffer ensures that pH remains stable throughout the demonstration. We add trypsin inhibitor to 2 of the vials. And add trypsin to 1 of the vials without trypsin inhibitor and 1 of the vials with. We then add a solution of ProAct to 1 of the vials containing soy. We also add it to the vial containing soy and trypsin inhibitor. We vigorously mix all 5 vials to ensure their contents are evenly distributed. We then incubate the vials for 10 minutes under conditions that closely mimic the conditions of monogastric small intestines, with a pH of 6.5 and temperature of 40 degrees Celsius. Finally, we centrifuge all 5 vials at high speed to separate the soluble material from the insoluble. First, let's look at the effect of ProAct on soy protein degradation. To the left is the control vial of soy, to which no protease has been added. In the middle is the vial of soy to which we added trypsin, and to the right is the soy to which we added ProAct. The blue-dyed proteins in the control vial remain intact and insoluble, leaving the buffer solution almost clear. But in the vials incubated with trypsin and ProAct, the solution is stained very blue. That's because the blue-dyed proteins were broken down and solubilized during incubation with trypsin and ProAct, allowing them to mix with the buffer. Next, let's compare how trypsin inhibitor impacts the effects of trypsin and ProAct. To the left is the control vial of soy and buffer. In the middle is the vial of soy to which we added trypsin, along with trypsin inhibitor. And to the right is the vial of soy to which we added ProAct, along with trypsin inhibitor. In both the control vial and the vial containing trypsin and trypsin inhibitor, the solution is almost clear, indicating that the blue-dyed proteins remain almost entirely intact and insoluble. So we can conclude that the trypsin inhibitor is blocking the effect of trypsin. By contrast, the solution in the vial containing ProAct and trypsin inhibitor is stained very blue. That's because, unlike trypsin, ProAct is not impacted by trypsin inhibitor. It has overcome the trypsin inhibitor to break down and solubilize the blue-dyed proteins. What we can conclude from this demonstration is that ProAct effectively breaks down soy proteins. We can also conclude that, unlike endogenous trypsin, ProAct can overcome the effect of trypsin inhibitors. Supplementing endogenous trypsin with ProAct in livestock diets improves performance through improved protein digestion and utilization. That allows for diets with less protein or soy protein of lower quality, leading to reduced feed costs. Improving protein digestibility also leads to less nitrogen in manure, resulting in lower nitrogen emissions.